Hi class, welcome to our Molecular Biology and Diagnostics Period 1 review, your comprehensive review, or what we call the discussion of some of the special topics that we were not able to discuss sa ating mga asynchronous lecture sa Period 1. This will serve as your reviewer and as a tool to help you or guide you for taking your Period 1 periodic examination. So yung mga lalabas na topics dito or discussion may or may not be included on your periodic examination. So first, let us discuss your electrophoresis, gel electrophoresis, walang kamatayang electrophoresis. So natatandaan ko may mga nabanggit ako na mga maling information, medyo nalito ako doon at first. Pero let me correct yung aking mga na-discuss na parts of the gel electrophoresis. Kasi nagkaroon ng confusion sa ating laboratory examination. So let us dissect each and every part of your electrophoresis machine. So number 1, tingin po tayo sa ating right side, we have your power supply. And then sa power supply, meron kang two types of wires. It represents your 2 electrodes. Meron lang silang main differentiation when it comes to positive or negative electrode. Pag sinabi natin electrode na black, it represents your negative electrode or what we call your cathode. Okay, let me correct myself. Cathode po yan. So yung red na wire, it represents your positive electrode or what we call your anode. So let me correct myself. Anode ito cathode. So itong mga to, this is what we call your sample wells. Dito natin pinupur ang ating samples together with your intercalating dyes. Sample wells ang tawag dyan. Hindi po direction of movement. May nakita ko nagsagot sa ating laboratory examination ng direction of movement. It is not a part of your electrophoresis, but rather the movement or the direction where your DNA will flow or follow. It is the migration. Okay? Next is what we call your buffer solution. Nandito siya banda. Okay? Your buffer solution serves as it carries the current and it protects the sample. Kasi nga buffer, meaning meron siyang pH, it neutralizes the pH kung saan nagkakaroon ng protection of the sample kasi nga merong iba't ibang pH yung sa may DNA and then yung mga pinupor na dyes. So the main job of your buffer is to protect the samples na nagmamigrate towards your positive electrode. Again, this is what we call the power supply, the negative electrode or what we call the cathode, the positive electrode or what we call your anode, and then your buffer solution. Huwag pong kalilimutan yan. So, tingnan po muna natin ang ating box on our left side. Your gel electrophoresis, its principles is that it is the migration of molecules based on charges or sizes. Depending on the charges, one will move from one region to another. So from the negative electrode, automatic your DNA will migrate towards the positive electrode. So ang inyong mga anions, since they are negatively charged, they will migrate towards the anode. So yung mga cations nyo naman, they will migrate towards the cathode. Since negatively charged ang mga DNA, automatically magmamigrate sila papunta sa positive electrode, which is the anode. I hope I made myself clear. Your cathode are negatively charged because it attracts cations, and then your anode, they are positively charged because it attracts your anions. Since the net charge of your DNA is negative, papunta po sila automatic sa inyong anode. And then the movement will be from cathode to anode. So for example, we have a question, which will travel faster, your 5 kb DNA or your 50 kb DNA? The answer for that will be your 5 kb. Since the bigger the DNA, the slower, and then the smaller the DNA, the faster. Next question, which will travel faster, your linear DNA or your circular DNA? The answer for that is your circular, because circular travels faster than the linear. And then next, na-encounter natin itong question na to for our laboratory examination. What does the phrase run to red mean? Run to red if it will be applied on a gel electrophoresis machine. What does the phrase run to red mean? Is it your DNA are moving toward the anode? Your DNA are moving toward the cathode? you run the gel until the first piece of DNA reaches the red line on the gel, or is it you need to use red dye when you load the gel for it to work? So I'll give you at least a minute to think of the answer and pwede kayong magsagot on your own. Okay, the answer for that will be letter A. Your DNA are moving toward the anode. Again, tandaan, ang ating anode meron siyang red na Y. It represents the positive electrode. So the phrase run to red, yung inyong mga DNA, papunta sila sa red, meaning papunta sila sa anode. Please do take note. Next, for the discussion proper of your module 1 to 9, halo-halo na siya. Hindi ko siya dinivide into specific topics. Basta kung ano lang yung madaanan at kailangan natin bigyan ng emphasis, sinama ko na siya dito. Again, for the central dogma of life, it will begin from your DNA and then will be transcribed to RNA and then will be translated to proteins. And the conversion of your RNA pabalik sa DNA, that is what we call your reverse transcription. So, almost lahat ng cells, dito sila dumadaan. Almost lahat. Dapat, dito sila dadaan. From DNA to RNA and then to proteins. Next, we have two branches of biochemistry specifically dealing with your DNA. So, naka-enroll kayo sa subject na yon, which is your cytogenetics and your molecular biology. But ano bang difference nung dalawang subject na yun at bakit kailangan pa silang paghiwalayin? Your cytogenetics deals with your DNA. Yes, it deals with DNA, but it focuses more on the chromosome. Okay, kung matatandaan si chromosome, it is a DNA package into structures that can be observed under microscope. It is where your DNA are being embedded. Sa cytogenetics, you're dealing more with your karyotyping, the Punnett square, puro ganon, and the diseases associated with the chromosome failures. Parang ganon. Whereas for your molecular biology, yes, we're dealing with DNA, but it is more involved in the individual nucleotide sequences. So, dinadissect natin siya on a molecular level. Mas tinitignan natin yung pagkakasunod-sunod or the sequences of the nucleotide to detect ano ba yung mga magagandang common molecular techniques that can be applied, and then yung mga diseases associated with the nucleotide sequences. Okay, yung mga mutation, dyan na siya papasok. Plus, your molecular biology is a pure science. Yes po, pure science siya. But if you apply molecular biology for research, diagnostic purposes, treatment of diseases, it is not anymore pure science. It becomes applied, and we can call that molecular medicine. Okay? Next, these are the common molecular techniques. As we all know, your polymerase chain reaction. In layman's term, your PCR is simply producing copies of DNA, similar to reproducing a page in a book of several times. Again, it is producing billions and billions of copies para ma-detect natin kung yung targeted DNA ba natin ay present doon sa sample na sinubject natin to test. Your PCR in vitro enzymatic application of your specific DNA sequence, the maximum size of which is 600 base pairs. It is the size of DNA fragment that can be copied. Next, your PCR requires a DNA polymerase enzyme that can withstand high temperature because PCR is done near boiling high temperature. So sa loob ng machine na to, meron siyang thermocycler, and then diba subjected siya into different temperatures. So tumataas, bumababa. Okay, so dapat it can withstand, yung mga DNA polymerase enzyme natin can withstand high temperature. Plus, your PCR is developed in 1983 by Dr. Karim Mullis, who received a Nobel Prize in Medicine and Physiology in 1993. Okay, for the summarization of your polymerase chain reaction, we have the three major processes wherein your denaturation, annealing, and extension are all involved. So, dissect natin siya isa-isa. Your denaturation of DNA, it is the separation of dsDNA into ssDNAs via denaturation. Pag sinabing denaturation, it is the breaking of the linkages. So, sa protein, ang binibreak natin doon yung mga peptides kapag ka naman sa inyong polymerase chain reaction having your denaturation, tinatanggal mo or pinagbe-break mo yung mga DNA strands. Okay? And then, ang ginagamit natin doon is yung heat. heat is used to denature your DNA strands. And then the temperature is about 95 degrees Celsius. Next, for the primer annealing, it introduces synthetic preformed primase, and the temperature is about 40 to 60 degrees. And lastly, the primer extension or the elongation, extended primers via synthetic heat-resistant DNA polymerase, usually from your thermus aquaticus or your TAC. Okay, TAQ is that it has a temperature of about 72 degrees Celsius. And then ano ba ang mga uses ng ating polymerase chain reaction? We have certain diseases can be detected using your PCR, yes, and it is used to investigate potentially defective DNA sequence, but not all diseases can be diagnosed. And then you can only diagnose a disease in which the specific mutational site is known. Amplified defective DNA can be amplified or compared to a normal DNA. So ano ba yung sinasabi natin na it can be used for diagnosis of a certain disease? Ito po siya. PCR in the diagnosis of disease is that expansion mutations of trinucleotide repeats such as in your Huntington's disease, Fragile X syndrome. So example, your Huntington's disease, your HD, has a normal gene of CAG repeats only. Yun yung pagkakasunod-sunod ng sequence ng kanyang mga nitrogenous bases. And then in Huntington's disease, CAG is expanded and becomes 5 repeats. So the cause of expansion is your slippage as is discussed on your genetics. Okay, ang principle ni PCR doon sa example natin na disease is we can amplify this DNA sequence. Next, we can use primers that can scan the site and using PCR amplify the DNA sequences. So para mas ma-visualize natin siya, ma-appreciate natin yung visualization ng Huntington's disease, ang gagawin natin, paparamihin natin yung nucleotide sample ng ating Huntington's disease genes. Okay, for example, ipaparamihin natin siya. And then 1 billion copies of the normal and expanded would be produced and then differentiate using your gel electrophoresis with the smaller fragments traveling far and the larger fragments traveling less. So ito po siya. Okay, please take note of the result that note that the migration patterns of normal versus expanded are different. Homozygous normal gene, which is shorter, travels farther while the expanded CAG homozygous expanded travel less and is found near point of the location. Size of the DNA fragment can also be estimated. In heterozygous, one gene is expanded while the other gene is normal. Whether patient will manifest the disease depends on whether the disease is autosomal dominant or autosomal recessive, plus your X-link or your autosomal. So doon sila nagkakaroon ng point of comparison doon sa homozygous normal and sa heterozygous expanded and homozygous expanded. Next, we have different types of your PCR. We have your touchdown, nested, inverse, multiplex, your real-time, and then your reverse transcription. For the real-time, during the process, you know you are amplifying. Okay, yung amplifying, the copying, or something as compared to the normal PCR, you only realize you amplified something at the end of your 4 hours. Sa real time, wala pang 4 hours, tapos mo na, nabasa mo na, nakita mo na sa graph yung amplification nung targeted mong DNA. Plus, we will not tackle only reverse the transcription. Okay? So yung reverse transcription, it is the production of your cDNA from mRNA. Again, it is the production of your cDNA from mRNA. In other words, we amplify your mRNA by converting it first to cDNA because mRNA is unstable. And then its uses includes the reverse transcriptase can be used to amplify expression of a gene. If a gene is active, it would produce mRNA that will only be amplified. So, ang mga requirements ng inyong reverse transcription PCR is the reverse transcriptase, the cDNA primers, the poly-A tail annealing primer, and then your TAC polymerase. So ito yung mga steps required for the reverse transcription. We have number 1, primer anneals to poly-A tail. The primer extended by the reverse transcriptase. the double-stranded cDNA is produced, the primers anneal to the cDNA, and lastly, the Taq polymerase amplifies your cDNA. So next question, in a PCR, a blank automatically changes temperatures and allows cycling to occur within a reaction vessel. Again, in PCR, a blank automatically changes temperatures and allows cycling to occur within a reaction vessel. Is it your thermocycler, your colorimetric assay, your primer, or your probe? Okay, so the answer is thermocycler. Tama po. Next question, which of the following is not required for DNA replication by PCR? Which of the following is not required for DNA replication by PCR? Is it your oligonucleotide primer, your DNA polymerase, your DNA ligase, or is it your deoxynucleotide? Okay, the correct answer is your DNA ligase. Hindi po siya required. Okay, for the DNA replication by PCR. So kung babalikan nyo yung mga requirements for the DNA PCR, yun po. Hindi kasama si DNA ligase. Next, it is the technique that makes your ssDNA from an RNA template. It is what we call, number 1, stranded displacement amplification. Is it your polymerase chain reaction? Is it your ligase chain reaction? Or is it your reverse transcription? So kung magbe-base tayo sa picture natin on our right side, makikita na natin ang sagot. It is what we call your reverse transcription. So meron kang RNA template and then ang goal mo is gawin mo siyang ssDNA. Okay, so pabalik. Okay, that is what we call your reverse transcription. So ito yung mga included pa din sa ating common molecular techniques that I wasn't able to elaborate on our period 1. So ito, bibigyan na natin siya ng focus. So paki-take down notes po. These are the blotting techniques. We have your 4 blotting techniques, but we will only discuss the southern blot since it is the basis for others. So yung western blot, northern blot, and southwestern blot, basically naka-anchor sila sa principle ni southern blot. Nagkaiba-iba lang sila sa kanilang analyte na ina-analyze. Okay? They are the same when it comes to the principle of the southern blot. Doon sila anchored. Nagkaiba-iba na lang sa kanilang analyte na ina-analyze. For the southern blot, okay, ang inaano natin dito ay later na lang. For the western blot, protein is being analyzed. For the northern blot, RNA is analyzed. And for the southwestern blot, DNA is analyzed. So para madaling tandaan, meron po tayong mnemonics dyan later on. So the same principle is applied for all of these 4. Difference only is what is being analyzed. Also for southern and northern, radioactive probe, pakitandaan, is utilized. Meanwhile, for your northern or your protein, we utilize radioactive antibody. Your southern blot is developed by Edward Southern of your Edinburgh University. It is where it is the analysis of DNA using complementary radioactive probes. Again, sa southern blot, it is for the analysis of DNA using complementary radioactive DNA probes. Its method is to isolate and identify the DNA in a complex mixture. It can locate particular fragments of DNA in a complex mixture and can quickly identify a gene or a gene segment within the genome. Okay, it is older because it was developed noong 1970s pa, but the PCR was only developed during the 90s. And then the formal name of the technique that is being currently used is DNA blot. So how is it done? Anong mechanism of your southern blot? Number 1, we wish to identify the gene 1. We have the fluorescent green. So ang pinaka-step nito is you will cut the DNA into manageable fragments using your restriction endonucleases. Again, you will cut the DNA into manageable fragments using your RE or what we call the restriction endonucleases. Number 2, arrange the DNA fragments according to size using the gel electrophoresis. So, subjected pa din po siya sa gel electrophoresis. Parang ganito yung itsura niya. Okay? Ikakat mo muna yung mga DNA fragments and then ilalagay natin siya sa ating gel electrophoresis machine. And then doon napapasok yung mga parang blotting paper. Okay, what we call, kaya nga siya blotting paper. Parang ganun yung kanyang mechanism. Next, highlighted here is is the one we're interested in, but we do not know yet that it is there. How do we know to identify it? This is where your probe comes in. So mahalaga si probe sa ating southern blotting or for the blotting techniques because next, it transfer the DNA from the gel into a piece of nitrocellulose by simply blotting. Para ka lang nag-blot nung tissue sa mukha mo kapag ka-oily ka na. So that is why it is called southern blot. The piece of paper will then be soaked into a solution that contains the radioactive probe. The radioactive probe is a piece of DNA that is complementary to the DNA you are trying to find. So when they are mixed together, it will bind for its match. At this point, the fragments are still invisible. This is where your X-ray photography comes in. Kaya nga po siya tinawag na radioactive DNA probes. This is the radioactive X-ray picture. Everything is clear except for this one. The DNA you are trying to locate visualized because of the radioactive probe. So para mas madaling tandaan, meron tayong mnemonics for the blotting techniques bago natin siya i-discuss further sa ating illustration on our next slide. This is what we call the snowdrop. Okay, snowdrop. Pag southern blot, southern S, it is analyzing the DNA. Okay? And then the northern blot, it analyzes the R, or what we call the RNA. Depende kung RNA virus or what siya. Basta kapag ka-RNA ang present sa kanya, ang gagamitin nating blotting technique is automatically northern blot. Next, for the western blot, ang ina-analyze natin dito is your proteins. So, yun po yung palatandaan. Kasi nga po, they have the same principles. Naka-anchor sila lahat kay southern blot. Nagkaiba-iba na lang sa kanilang ina-analyze. Okay? For the southern blot, DNA, northern RNA, western protein. So, this is the visualization or the illustration of how your southern blot works. So, we have your restriction-digested genomic DNA kung saan ikakat na natin into manageable fragments ang inyong DNA and then will be subjected to electrophoresis. So, depending on the size and on the charge, it will migrate. Okay? Next, your southern transfer, kung nasaan nandoon na yung inyong nitrocellulose paper, parang ganon, ibablot nyo na siya. Okay, and then your radioactive probe, meron pa siyang mangyayaring washing. The probe hybridized to restriction fragment and then it will expose into X-ray film and then tsaka na natin siya babasahin as X-ray film or X-ray picture film. Parang ganon. So these are the applications of your southern blotting. The gene discovery, mapping, evolution, and fingerprinting and diagnostic and forensic. So this can be used for yung mga SOCO or yung mga forensic investigation natin. Yung DNA profiling or DNA fingerprinting, applicable yun. Doon ina-apply ang southern blotting. Definitive test to ensure that a particular section of DNA of known genetic sequence has been successfully incorporated into the genome of the host organism. Also, it can detect mutation as well. So, dito muna tayo sa DNA fingerprinting. In the DNA fingerprinting, we analyze fragments called your VNTR. Okay, your VNTR stands for the variable number of tandem repeats. Okay, your junk DNA, non-coding, non-transcribed, and then meron tayong tinatawag na short tandem repeats or your STR. The tandem repeats, repeating yung ganyan, variable number varies in the number across the individuals within own DNA. It is unique for each person and can therefore be used as basis for determining identify and individuality. And then a copy of VNTR of an individual comes from the father and the other copy is coming from the mother. It is great for establishing your paternity. Ginagamit siya sa mga paternity testing. So, example, ito po siya. Paano ba ginagawa ang DNA fingerprinting? Paano mo siya masasabi na ito yung mother, ito yung father by basically looking lang doon sa pattern ng tinatawag nating mga DNA bands. So for example, an individual, number one, first we cut using yung ating RE. Okay, the DNA fragments will be produced of varying sizes. Size will determine the size of your V and TR. Next step is we will arrange the fragments from smallest to largest using your gel electrophoresis. And then the banding pattern, which is invisible, will be produced. Number three step is the transfer into the paper by blotting, and then the probes for the VNTR will be utilized. So dito na papasok yung mga blotting techniques natin or your southern blot. And then the probes will bind to the DNA fragments and then to take an X-ray film. A banding pattern that is unique for you is then made. So paano siya naging applicable sa establishing of a paternity test or application for the forensic purposes? For example, ito siya. Okay? So, balikan na lang natin yung slide kanina. For example, ito yung blood stain na nakita sa isang crime scene. So, sino ba doon? Si suspect 1, 2, 3, and 4, yung merong matching band patterns doon sa blood stain na nakita natin sa crime scene. So, pakisagutan. I will give you a minute to think. Sino ba yung pinaka-close yung banding pattern niya and then match doon sa blood stain? Okay, tama po. Si suspect number 3. Si suspect number 3, o diba? Halos same sila ng pattern nung pinaka parang control or yung blood stain coming from the real suspect. So, si suspect number 3. Next naman, for example, meron tayong soldier and then gusto niya malaman kung sino yung parents niya. So, yung pattern nun, depending kasi, di ba, 50% of your DNA coming from your mother and 50% coming from the father. So, yung banding pattern niya, meron ka ng dalawang basis. So, depende kung pattern siya sa inyong mother or dun sa father ng soldier pala rather. So, sinong parents ba? Si A, B, parents C, D, E, F, or GH. So, paki-pattern, tingnan kung kanino yung best match or best fitted doon sa soldier depending sa bands niya. Okay, tama po. Ang sagot ay parent C and your parent D. Okay, so itong band na to coming from the D, let's say father. Ito naman, itong pattern niya na to. Itong pattern na to kamatch nitong nasa soldier. And so on. Ganun po siya. That's how your DNA fingerprinting works. And then, for example, apply natin siya sa forensic purposes. Then the ladder, the DNA of known size or your reference point for comparison and the estimation of bands, molecular weight, and size. For the assessment, suspect is the suspect 1 because the banding pattern is the same as the sperm specimen. So, tingnan natin. So, ito yung ladder. Kunwari, kanino siya pinaka-closely related or same ng pattern depending si suspect 1 daw. Okay, for example naman, in establishing the paternity, your daughter number 1, okay, if you will look closely, bands that matches the moms and the bands are matches the dads. So, si daughter number 1, check natin. Same ba sila kay mom? Yes, sa dad. Okay. Okay, so legitimate child siya. Next, your daughter number 2, bands that matches the mom but no bands that matches the dad. Daughter number 2, puro kay mom nakuha, wala siyang band na same as pattern kay dad. So the daughter with another guy. She is the daughter from another guy. Okay, son number 2 is a legitimate child of the mom and dad because if you will look closely, okay, tama naman. They have the same pattern aligned to the mom and the dads. And the son number 3, tingnan natin. Wala namang son number 3. So, eliminate na natin yan. For the last example, Thomas Jeffrey's story is the basis of for males is the DNA from the Y chromosome and the basis for females is the DNA mitochondria. So, question. This is a modified true or false. Ito, ano lang to, parang exercise for the upcoming periodic exam. Letter A, statement number 1. Southern blots are produced when a radioactive DNA probe hybridizes with DNA on a nitrocellulose blot of a gel. It is often used to detect DNA mutations. Is it true or is it false? Statement letter B. Northern blots are produced when a radioactive DNA probe hybridizes with RNA on a nitrocellulose blot of a gel. Ano po ang tamang sagot? So you can pause this video if you need time to think of the answer. But if you know already, pwede nyo na i-proceed. The answer is letter C. Both statements are true. So now we're done discussing the common molecular techniques as well as the, balikan lang natin, nakalimutan ko siya. Common molecular techniques and yung mga konting about electrophoresis na kinlarify, the PCR, and how your blotting techniques work. So now, let us proceed with the discussion of the nucleic acid. So hindi ko na siya dinisect isa-isa as to per module kasi madadaanan na siya dito sa ating overview or review lang kasi dapat na-review nyo na siya or naaral nyo na yung lecture video provided prior to this discussion. So for the nucleic acids, dadaanan nito yung DNA and RNA, and ang pinaka-last part of the discussion will be about the mutations. So your nucleic acids, the basic structure of nucleic acids is that nucleic acid is a polynucleotide, a linear polymer of nucleotides made up of 3 components. Alam na natin yung tatlong basic components of your nucleic acid or your nucleotides. We have your nitrogenous bases, your 5-carbon sugar, and then your phosphate groups. Okay, ewan ko na lang, makalimutan nyo pa to. For the nitrogenous heterocyclic bases, we have your purines and then your pyrimidines. It is attached to the 1 carbon of the sugar by a N-glycosidic bond. Again, ang glycosidic bond, it can be referred to the connection or the attachment of your sugar to another sugar or sugar to another other groups pa. Pwede siyang i-attach sa ibang phosphate groups or to other groups pa. Hindi lang siya limited to the attachment of sugar to another sugar. Next, your purines have a double ring and include your adenine and guanine. Adenine and guanine, they are under your purines because it has double rings. Kapag single ring naman, it can be cytosine, your thymine, or uracil in case of an RNA. Next, your both DNA and RNA contain your adenine, guanine, and cytosine. Thymine is only found in the DNA, meanwhile uracil is only found in the RNA. This is just a review. And then your cyclic 5-carbon sugar residue. RNA contains 5 ribose sugar, which OH groups bound to its 2' and 5 carbons. DNA contains deoxyribose sugar. Yun yung kanilang main difference. Deoxyribose is identical to ribose except your OH group at the 2' position. Again, saan po? Sa 2' position ng inyong pentose sugar. 1, 2, 3, 4, and 5. Basta. Dito siya. 1, 2. Dito. Has been replaced with the H atom. So yun lang yung kanilang main difference. And the phosphate group is attached to the 5' carbon of the sugar by a phosphodiester bond. And it is responsible for the strong negative charge of both nucleotides and nucleic acid. Okay, tandaan. Nasa phosphate group ang dahilan kung bakit negatively charged ang ating mga DNA. The nucleotides are joined to one another by a second phosphodiester bond or your phosphodiester bond between 5-phosphate of one nucleotide and the 3-OH group of the adjacent nucleotide. This arrangement is what we call your phosphodiester bond. Yun po yung tawag na. Next, For a review or exercise question, which of the following statements indicate the direction of the DNA or RNA polymerase synthesize a new strand of RNA or DNA? Yung direction daw yung tinatanong dito. Is it from the second prime end to the 3' end? Is it from the 5' end to 3' end? Is it from the 3' end to 5' end? Or is it from the 5' end to the 2' end? Pag tinanong yung direction, indicate the direction. Okay? Synthesis. Synthesis po, ang sagot po dyan, 5' to 3'. Baka may ma-encounter din kayo na same question, pero ang tanong, reading. Okay? Ang reading naman, 3' to 5'. Pero pag synthesis ang pinag-uusapan, 5' to 3' end. Automatic po yun. Next, for the physical and chemical structure of the DNA, we have your base pairing according to the Chargaff's rule. Base composition of DNA varies from organism to organism. However, A always pairs with T. Okay, yung ating palatandaan na A and T. Laging sila yung magka-partner. Or U in case of your RNA. And G always pairs with your C. Okay, G and C, parati po sila magka-partner. This is termed as complementary base pairing according to your Chargaff's rule in DNA. The number of adenines equals to the number of thymidines and the number of guanines are equals to the numbers of your cytosine. Parati sila. Kasi once na na-predict mo na yung numbers of your adenine, o ibig sabihin pareho na din doon sa thymidine or thymine of the antiparallel. Okay, in a double-stranded DNA molecule, the concentration of purines are always equal to the concentration of your pyrimidine. Please do take note. Another one is the structure of your nucleotides or your DNA and RNA or your nucleic acid. It is Watson and Crick on 1953 described the DNA as two polynucleotide strands coiled about one another to form double-stranded helix. And then the sugar phosphate backbones of each strand from the outer edge. Kaya sila naging backbone kasi yung sugar plus phosphate, kung mapapansin nyo, nandun sila sa labas, outer part siya. Kaya siya naging backbone. Parang backbone ng pagka-double-stranded helix ng anti-parallel na inyong DNA or RNA. Of the molecule and bases are in the internal central core. Pag yung mga nitrogenous bases, nandun po yan sa loob. Okay, they are being joined by the hydrogen bonds. Pero yung mga sugar and phosphate, backbone sila. Sila yung pinaka-backbone ng ating mga nucleic acids. Each base in one strand is hydrogen bonded to a complementary base in the other strand. So, para mas ma-visualize nyo siya ng maigi, meron po tayong PowerPoint provided and meron din tayong lecture video provided dissecting each parts of the DNA. Parang ano siya, module number 2. Okay, balikan nyo doon para mas maganda yung visualization niyo. Ito kasi review na lang. Okay po, which forms the purine and pyrimidine base pairs or your BP. The example, AT and GC. 2 hydrogen bonds between your AT. This is just a review. Pagka naman 3 hydrogen bonds, G and C on your guanine and your cytosine. And then at physiological temperature, the DNA base pairs are stable. However, they can break and reform rapidly. And then, kasi kung matatandaan nyo, these adenine and thymine, they are being joined together by hydrogen bonds. E ano bang ginagawa ng mga denaturing reagents ninyo or yung ano bang property ng inyong mga denaturation chemicals? That is to break the hydrogen bond. Yun yung goal niya. And then a DNA helix has 2 external grooves. We have what we call your major groove, where various protein molecules bind to DNA, and the minor groove. So yun lang naman yung kanyang job. Basically, sa mga binding of the protein molecules. Kaya mahalaga yung mga major groove and minor groove natin. Next, for the complementary strands, due to base pairing rules, the 2 strands are complementary. For example, if one strand has a sequence of 5' to 3' na may ganitong sequence, dapat alam mo na kung paano siya, ano yung katapat niya doon sa other strands. Okay, kung titignan mo pa lang. Okay, sagot dyan, 3, ito, G, ito, si C. Okay, A and T, T and A and T, C and G, C and G. Ganun po siya. Complementary nga sila. Always partner si A and T, si G, at si C. Next, for the DNA replication. The DNA replication is a process in which, in which genetic information is transferred from parent to another cells. It requires energy to unwind the helix and disrupt the hydrogen bonds. Ano po yung enzyme na nag-unwind doon sa ating helix? That is what we call your helicase. It requires ATP. Yes, it requires ATP. Sa DNA replication, meron tayong tinatawag na template strand, for example. Okay, itong mga template mo from the old DNA, ang goal nila makagawa ng panibagong DNA by making or parang magre-reproduce sila ng daughter strands or daughter cells. Okay, from the template sila yung old, gagawa sila ng panibago or what we call yung mga mas younger cells or yung younger template. Proofreading and repair systems exist to minimize the replication errors. However, mistakes do sometimes occur and on occasion can be expressed as mutant phenotypes. Base changes occur resulting in mutations. Yun yung mga tinatawag natin mutations. Kapag patuloy kasi na nagre-replicate yung inyong mga DNA natin, meron tayong tinatawag na mga proofreading and repair systems para makita or ma-double check or ma-quality control kung may mga naibaba doon sa ating mga base pairings. But however, may mga nakakalusot at nag-evolve siya. At doon nagpapasok yung mga tinatawag nating mutations, which will be discussed further sa ating latter part ng ating review. Next, these mutations do have an effect producing polymorphisms. Often mutations have detrimental effects. However, some mutations result in a selective advantage for the organism that is the basis for evolution. So lahat naman tayo dumaan tayo sa mutation. Lahat tayo nag-umpisa sa mutation na kung saan, kunwari, parang sa chicken na naging dinosaur. Parang naman ganun. Kasi nga, through the years, nag-evolve and then through dun sa replication and replication of the DNA, merong mga nagagawa or may napoproduce na polymorphism. The synthesis of each nucleotide chain only occur in the 5' to 3' direction. One strand is synthesized continuously whereas the other strand is synthesized discontinuously. Ito yung tinatawag nating Okazaki fragments doon sa tinatawag natin mga strands that must be ligated together by the enzyme ligase. Each parental DNA strand serves as a template to create a complementary daughter strand. As replication proceeds, the parental double helix unwinds by the action of enzyme or what we call the helicases in prokaryotes and topoisomerase in your eukaryotes. Polymerization of DNA is catalyzed by enzymes called your DNA polymerases. Yung mga topoisomerase, kung natatandaan, sila yung nagpa-prevent ng mga supercoiling as the helicases unwinds your DNA strands. Doon sa dulo, merong tinatawag na mga topoisomerase for the eukaryotes. Kapag sa prokaryotes, yun yung tinatawag natin mga DNA gyrase. And then, as a new strand is formed, it is hydrogen bonded to its parental template. Each new double helix consists of one parental strand and one newly formed daughter strand. So, going back lang, paki-review po na maigi yung DNA replication kasi hindi lang siya ganito kasimple. Complex siya in a way na sobrang daming enzymes involved. Tapos yung buong process niya is medyo complex nga po siya. So, balikan po ninyo yung lecture video natin for the DNA replication. Ito kasi, overview na lang kasi review na lang siya. Pinapaalala ko lang na ito yung paano mangyari, paano yung ganap ng each enzymes. Kasi review na lang siya. Pero kung gusto nyo mas malaman siya into details, pakibalikan po yung lecture video natin. Kung mapapansin nyo kasi mabilis lang talaga yung discussion natin dito. Okay? So going back, let us discuss your RNA. RNA is a sugar moiety is ribose rather than 2' deoxyribose DNA. The pyrimidine component is uracil. Please do take note. And it is existed as single strand. Kaya siya, single-stranded DNA. Parang RNA po siya. And then to distinguish an mRNA, it has a cap and a tail. mRNA stands for your messenger RNA. And then you'll be needing a cap and a tail. After you synthesize an RNA through transcription, this will undergo post-transcriptional processing. So, na-discuss po ata to sa ating lecture video. Again, pakibalikan na lang. So, these are the roles of different kinds of your RNA. Meron tayong tinatawag na tRNA, your transfer RNA. It has a small size and functions as a transport of amino acids to site of protein synthesis. And the extent of hydrogen bonding is considerable. Next type of your RNA is what we call your ribosomal RNA or your rRNA. It has different or several kinds of sizes depending talaga siya. And its function is it combines with proteins to form ribosomes and then the site of protein synthesis. And the extent of hydrogen bonding is considerable. And lastly, your mRNA or your messenger RNA, ito yung most commonly na nai-encounter nyo or naririnig nyo sa discussion. It has varying sizes and its main function is it directs amino acid sequence of the proteins and has a very little extent of hydrogen bonding. So kung tatanungin ano po yung pinakamaliit na size ng RNA, that is what we call your transfer RNA. Next, for the RNA overview, the most RNA molecules are single-stranded. Again, most RNA molecules are single-stranded. However, RNA readily forms secondary structure. So generally, RNA is environmentally labile and easily degraded. So the types of the RNA includes your rRNA or your ribosomal RNA, your mRNA or messenger RNA, and tRNA or your transfer RNA, and we have other types of RNA. So let us discuss each and one of the RNA. Your rRNA makes up 80 to 90% of the total RNA in a cell. It is part of the ribosomes and is involved in the translation of your mRNA into proteins. Yun yung rRNA ninyo. Kaya nga po siya ribosomal RNA. Next, for the messenger RNA, it makes up about 2.5 to 5% of total RNA in a cell. It is an intermediate between the genetic code in DNA and the protein product. mRNA is read by ribosomes to produce your proteins. In eukaryotes, transcription of DNA forms from a pre-mRNA molecule with both introns and then your exons. This molecule is referred to as heteronuclear RNA. The introns are removed and then the exons are joined together. Further processing into mature mRNA includes addition of a 5' methylguanine cap and a poly-A. Ito yun, si poly-A tail up to 200 adenylate nucleotides in the 3-OH terminus. Next, your transfer RNA reads. It reads again your mRNA triplets and brings the appropriate amino acid to the ribosomal polypeptide synthesis. There is at least one tRNA for each amino acid. And lastly, meron tayong small nuclear RNA. It is involved in the removal of the introns, and your small and micro RNA, yun yung mga ito. These are involved in cellular processes, but mainly ito talaga yung mga main types of your RNA na malaki yung involvement nila for the translation from RNA up until your proteins na siya. So the genetic information is usually stored as DNA, but it can be stored as RNA as well. RNA and DNA is a polynucleotide or polymer made up of nucleotides or your monomer. The nucleotides contain sugar, bases, and the phosphate. Okay, review na lang po ito. Si phosphate group, again, it gives off the negative charge to the DNA and RNA. Hindi lang po si DNA ang may negative charge, si RNA din siya dahil nga sa kanilang phosphate group. And then the central dogma of life, saan nga ulit nag-uumpisa ang DNA replication sa ang part of your cell mitosis? Doon po siya sa S-phase of the mitosis. Okay, pakitandaan S-phase. The genes are expressed through transcription or translation. The products of transcription, we have your rRNA, tRNA, and mRNA. Not all the products of transcription are translated to a protein. Only mRNA becomes protein. Tandaan yung mRNA lang yung nagiging protein. And then these proteins are very important as they are being translated from RNA because these proteins or amino acids build most of our cells, lahat, pati yung mga organs, lahat yun. Marami siyang functions ang ating protein sa ating katawan. So the rRNA is associated with ribosomes. Your tRNA transfers amino acids to the site of protein synthesis, and your mRNA directs the amino acid sequence. Okay, the messenger of genetic information and acts as a template for translation. So pinakamalaki talagang ambag dyan, si mRNA. And this is the secondary structure of your DNA. That is just a review. So pakitingnan po yung picture natin on our right side. Meron tayong tinatawag na stop codons. Meron tayong UGA, UAA, and UAG. So ano po bang ganap? Ano pong meron sa ating mga codons? These codons signals the end of the polypeptide chain and during translation. Okay, again, these codons signal the end of the polypeptide chain during the translation and they do not code for any of the amino acids. So kung matatandaan yung mga codons na to, yung tigtatatlong base pairings, nitrogenous base pairings niya, meron po silang kinocode na amino acid. So kung babalikan, meron po silang amino acid. But these three, they do not code for any of the amino acids. Sila talaga yung stop codons natin na kung saan masasabi natin na tapos na yung translation. So meron tayong tatlo. Si UGA, you go away. Ang palatandaan natin dyan, you are away, URA, and UAG, you are gone. Ibig sabihin, stop na po. Bakit po siya mahalaga? Discuss natin sa susunod na slide. So again, review lang, your single-stranded DNA or your DNA can go through denaturation. It basically destroys the hydrogen bonds. Saan po nahahanap yung mga hydrogen bonds? Doon sa loob, meron tayong mga base pairs dyan, nitrogenous base pairs, and they are being connected by these hydrogen bonds. So they are being denatured. If you cut the DNA from one fragment to two fragments, dinestroy mo yung kanilang phosphodiester bond. And if you cut the sugar from the bases, you destroy the N-glycosidic bond. If you remove the phosphate, you are basically destroying the phosphoester bond. And this is the structural hierarchy of your DNA and your RNA. For the last part of our comprehensive review or the special topics, this will be about the mutations. So mutations basically means that it has something to do or changes that has something to do with your DNA. So from the beginning of time, meron na talaga, existent na talaga si mutation. And for me, mutation is inevitable. Sa lahat ng cells, mutations talaga may or might occur. So evident naman siya doon sa virus that is still going around, yung mutations. Naririnig niyo siya sa mga news. Kasi nga po, mutations talaga, it is inevitable and it is going on for the longest period of time. And ano ba yung mga nangyayari sa ating mutation? So mutations, it is the changes in the nucleotide or base sequence of your DNA. Again, changes in the nucleotide base sequence of the DNA. It may occur due to the errors in DNA replication or due to the impacts of chemicals or radiation to the DNA molecule. And the mutation may result in coding sequence for new amino acid in proteins or not. It may be, it is heritable. Any change in the sequence of DNA base pairs that is permanent. Pag nagkaroon ng permanent changes, automatically yung unang change doon sa DNA, yung mga susunod na DNA replication, makukuha na niya yung template, diba? Sa DNA replication, yung template of the old or parental strand, mamamana na siya ng mga daughter cells or ng mga daughter strands. Yun na, sunod-sunod na yun. It arises by chance. And then to be considered a mutation, the change in the base pair sequence must not be a result of your recombination. Next, What are mutations? These are heritable changes in the nucleotide sequence of a given DNA. Nagkaroon po ng mga changes doon sa nucleotide sequence ninyo. Kasi nga, each nucleotide sequence codes for anything. The substitution or insertion or deletion of one or more nucleotides. Depending na lang kung nagkaroon ng substitution, insertion, or deletion. Mayroong tatlong pwedeng mangyari doon sa nucleotide sequence kaya nagkakaroon ng mutations. It could affect or not a given phenotype. It is the major basis of diversity among organisms. It is the raw material of evolution. It is caused mostly by mutagenic agents and can be repaired by the body in normal conditions. And then these are the classifications of mutations. Meron tayong tinatawag na dalawa based on the nature of their occurrence. It can be spontaneous, meaning it arise in the absence of a known mutagen. Okay? May nangyari dyan na strand slippage. And meron naman tayong tinatawag na induced mutations. Meron tayong presence of mutagens or environmental agents. For example, yan yung mga exposure nyo sa radiation, yung pagka hindi gumagamit ng mga sunblock, parang ganon, it can lead to skin cancer. Okay po, these are some examples of the mutagens or mutation, sorry. Yung paggamit naman ng smoking cigarette, may na-embed sa DNA ng mga parental strands yun, and then it can lead to mutations. Kasi nga may na-introduce kang mutagens doon sa tinatawag mo na yun. May mga extrinsic factors that influence the mutation. Next is based on the cell type where it occurs, depending if it's somatic or gametic. Next, this is the molecular basis of mutation. We have 3. Yung substitution, for example, meron kang coding of the genes dito, may nasa substitute na letter. Okay, there's a change in the one letter, substitution yung tawag doon. For example, in layman's term naman, the deletion, meron kang code dito of the genes, may na-delete na isang letter, nawala yung C. Okay, that is what we call your deletion. Next naman, from the parental strand, and the cat saw the dog, nagkaroon ng insertion ng isang letter, nag-gain siya ng one letter, ang tawag doon insertion. So, from the parental strand, nagkaroon ng, imbis na the cat saw the dog, for example, the bat saw the dog, iba-iba na yung letter, substitution yun. Pag may naburang one letter, that is what we call your deletion. Pagka naman may na-insert or na-add na isang letter, that is what we call insertion. So, the point mutations affect the single sites of the DNA. The substitution of one base for another. Kasi nga, pagka naiba na yung isang letter doon sa inyong DNA, yung susunod na code nun, mag-iiba-iba na siya di ba? So, kaya nagkaroon ng mutation. Next, the deletion or addition of a single base. And then the deletion or addition of a small number of bases. If purine, A or G, or pyrimidine, T or C, substitute for itself, it is the transition substitution. If the purine substitute for pyrimidine or vice versa, that is what we call the transversion substitution. So, basically, kaya lang naman nagkakaroon ng mutations kasi as you make a lot and lots of copies of the DNA, so sobrang daming copies nun, at some point, nagkakamali. Okay, yung code nung isang amino acid doon, nagkakamali yung code. So, yung pagkakamaling code ng amino acid na yon, basically, mare-reproduce siya. Okay, magiging parental strand siya. Habang nagiging parental strand siya, nagkakaroon siya ng daughter strand at nauulit-ulit na. Iba na yung coding niya. So, parang COVID virus siya na same pa din sila na as virus pero merong naiba doon sa kanya mga protein spikes. Parang ganon. And then the results of point mutations, this is what we call your silent mutations, your missense mutations, and then your nonsense mutations. So these are the sample outcomes of the DNA code. So for the silent mutation, due to the redundancy of the genetic code, no change in amino acid sequence is produced. So ito po siya, amino acid sequence. Di ba, binabasa sa mga machine natin yan yung amino acid sequence to know kung anong type of organism ba yung binabasa natin doon. Next, for the missense mutation produces a change in the amino acid sequence. Naririnig nyo ba yon, yung amino acid sequence in the protein product? It may be histidine okay, yung histidine, imbis na arginine, may be changed function of protein or may not. So may mabago man isa sa amino acid sequence yan, mababago na yung kanyang reading, mababago na yung kanyang function, mababago na yung paano nag-work ang isang cell. Next, for the nonsense mutation produces a stop codon. Again, ano ba yung stop codon natin? Dapat kasi meron lang point kung saan mag-o-occur si stop codon. Okay? During the translation. Kunwari, dito pa dapat si stop codon. Pero pagka nagkaroon ka ng nonsense mutation, nandun pa lang sa kalagitnaan ng translation, magkakaroon na ng stop, which is not a good thing. Stop codon within the mRNA transcript leading to a truncated protein. How short the protein product may depends on where the stop codon was produced within the mRNA transcript. So yes, meron po dapat stop codon but not in the middle, hindi doon sa mRNA template. At some point, a-appear siya but not on the mRNA transcript. So nagkakaroon ng changes, nagiging short yung proteins, yung amino acid sequence, and nagtutuloy-tuloy na po siya. Parang domino effect na siya. And then, how mutations causes diseases. For example, yung cystic fibrosis mo. Meron kang missing amino acid. So, ang tendency, signs and symptoms niya for the phenotype, it will occur. The patient will suffer from frequent lung infection. And then, same dun sa ating mga different diseases. So number 2, the chromosomal mutations change the structure of the whole chromosomes. Doon sa kanina, point mutation lang, meron lang point kung saan nababago ng deletion, merong insertion, merong substitution of an amino acid. Dito, buong chromosomes na ang nabago. Chromosomal mutations are more extensive, altering the entire chromosomal structure. These kinds of mutations occur through deletions, duplications, inversions, and then your translocations. So the deletion to chromosome, marami tayong mga chromosomal disease that will be discussed on period 2 na, but I think you were able to discuss it sa inyong cytogenetics pag nagkaroon ng mga chromosomal diseases or defections. If too much information is lost, kasi nga naman buong chromosomes na yun, sobrang daming DNA na yung nawala kasi nga chromosomal deletion. It may be fatal to the organism and may result in early death. So ito po siya. Parang example natin si normal hemoglobin pag sobrang dami na ng mutation pag sa chromosome na. 'Di ba? Yung sickle cell hemoglobin, it can be fatal kasi ang daming signs and symptoms na maaaring ma-record or mangyari kay patient that can lead to fatality. Next naman, ito din siya. Basically, parang normal gene, pag nagkaroon ng substitution and then the missing, ang difference lang is sa chromosome siya nangyari. Hindi siya doon sa nucleotide-based sequencing pa lang. So, ito siya. Naka-chromosome changes can be small or large, depending. And then the duplications within chromosome is its effect of base duplications depending on the location within the chromosome, whether or not duplication resides in coding or non-coding region of the DNA. And then inversions with chromosomes, ito po siya, inverted na. And that concludes our review or the special topics for your molecular biology and diagnostics. And these are some of the tips that I can give before your periodic examination. Number 1, read the whole module or book first because it contains all the explanations and then you have to take notes. Meron tayong provided na mga PDF copies of your lectures, of your topics. Meron tayong PDF doon sa ating Google Classroom. Pakibasa po siya kasi na-explain ko siya sa ating lecture but yung mga thorough discussion meron doon. And then next, go back to your notes and watch the materials provided. Try explaining it to yourself and see if you have the same understanding with the lecturer. And then avoid distractions. Do not cram. And then rest and then study again. Huwag tuloy-tuloy. Pwede namang mag-rest. pero mas madami yung rest kaysa sa studying time. And there is no shortcut. Okay? Kaya po ako nagsabi na walang shortcut kasi basahin nyo muna yung libro. Then, take down notes. And then, tsaka nyo panoorin yung mga lecture videos provided. And then, try explaining it to yourself. Again, wala pong shortcut sa lahat ng yan. And if you have questions or clarifications, do not hesitate to contact me. So, that's it. Thank you.